Good afternoon, everybody. Let me welcome you to the CRTM monthly lecture series, the first lecture of this year, both in presence here as well as those who are joining online. And good afternoon to everybody. First of all, let me talk a few words about CRTM. Center for Research in Tropical Medicine has been existing in this faculty from 1990s, and we actually relaunched our activities from 2014 with the committee and did 2 international conferences. Then during the COVID time, we had to actually hold our monthly activities, but we published one tropical medicine book, this one in 2020. So those were some CRTM activities. And this year, with the new committee, we are restarting our activities, and I wish to thank our coordinator, Dr. Chamra Dalugama, for taking a lot of effort to organize these activities and continue CRTM activities. So today we are gathered here with the first lecture, and our committee member, Professor Veranja Liyanapathirana, agreed to do the first lecture. So in a little while we will listen to her. And to give an introduction about Veranja, I don't think I need to introduce her because she's a well-known and reputed microbiologist, virologist in our country. So I don't want to talk about her microbiology achievements, but I'd like to talk a few words about Veranja's hidden talents. Right? So it was about 10 years ago. I received an email from my collaborator, CDC Atlanta, Professor Gigri Dash. He has sent me a journal page, that's the emerging infectious disease, and he has inquired, Sam, do you know this lady? Then I had a glance there, and it was a nice set of verses, right? Poems, right? And it was heading was OD to bugs or something, right? It was very interesting. So Veranja has actually showed her talent in a well-reputed international journal, right? I think she has been continuing that talent. Okay, without taking much time, over to you, Veranja.
Thank you, sir, for the kind introduction, and thank you, CRTM committee, for inviting me to do the first talk. And without further ado, let me start the talk. If Mr. Taranga can help me to share the screen. Yes. Yeah. Right. Thank you, Mr. Taranga. So the topic I chose for today is the evolving role of the microbiome in human health and disease. Just to start with a few disclaimers, this is actually not directly related to tropical medicine, but I'll make a connection towards the end of the talk. I have only worked with the microbiome for 7 months, so it's mostly based on reading than on my own experience, except for a couple of things. And I've, of course, received funding from the industry for a non-related work. So this is what I will be going through in the lecture, the changing concepts from normal flora to the microbiome, the evolving definitions of the microbiome, the role of microbiome in maintaining health, disease associations, therapeutic applications, and novel concepts when it comes to the microbiome research and microbiome. So when most of us were studying microbiology, either for medicine or for a related study, we learned about normal flora than about microbiome. So normal flora was the initial concept that we learned in the time when the characterization of microorganisms was basically based on aerobic bacterial cultures. So during these times, we knew that there were certain parts on the human body or in the human body that had certain bacteria. And then, of course, there were other parts like the deeper tissue, the lungs, the brain, the blood, which were traditionally considered as the sterile sites. Now, we also learned about a couple of advantages and disadvantages of these normal flora. those of who underwent the medical curriculum, of course, memorize these for third MBBS examination. Advantages being prevention of colonization by pathogen, synthesis of vitamin. Of course, even back then we were talking about how the gut microbiome, gut normal flora is important in the development of the immune system. And of course, if the normal flora enters to a site where they are not normally found, they can cause infections. And as microbiologists, we always talk about normal flora interfering with lab diagnostics and if the skin asepsis is not properly done. So from these times in the late 80s and all, technology advanced. It advanced very, very rapidly, mostly towards the latter parts of this century. From culture, we moved on to molecular-based testing, and particularly towards when next-generation sequencing was introduced in the mid-2000s. This is where you can just put a bit of sample into a sequencing machine. You can take it as a black box. Of course, there are some lab techniques involved, but you take the human sample, extract the DNA and RNA, put it in. It will sequence the human genomic material that is there as well as all the microbial genomic material that is there. Then with bioinformatics, we can actually extract out the human and the microbiological genomic material that is there. So when this technology evolved, we found that instead of just looking at culturable bacteria, that there is this hidden universe inside and on our bodies, which is composed of a multitude of bacteria, viruses, parasites, fungi, all the microorganisms. So this actually moved the concept of normal flora towards the concept of microbiome. So originally, as I mentioned earlier, people were using sequencing technology. So therefore, the earlier definition of microbiome is actually the microbial DNA that is there on a given site at a given time. Now, I am focusing on the human microbiome, but of course, the same thing is applied for animals, for plants, and everything. Now, then, afterwards, because technology evolved to a certain extent from there also, we moved from just calling the collection of genomic material as microbiome towards, looking at the actual microorganisms at a given time, at a given site, considering both the microorganisms that are there all the time and the transient visitors that we get, we started to call them together as the microbiome. At the time, the technology evolved further and our conceptualization of the microbiome also evolved further. Now we sort of encompass not just the microorganisms, but their products, their proteins, the substances they secrete, of course their genomes, and also the surrounding environment together as the microbiome. So when you're reading about microbiome, all those three definitions are there depending on, of course, the site where we read. So as mentioned earlier, it was the evolution of the technology or the development of technology that drove the microbiome research. So we, of course, had the sequencing technologies that evolved initially. Even with the sequencing, we were characterizing only the bacteria based on the 16S bacterial gene, which is universal to all the bacteria, and you sequence and identify the different bacteria there. Then the technology moved to what we call metagenomic studies, where we actually don't need to know what is there. We don't target for a specific pathogen. We sequence all the microbial nucleic acids there, and using bioinformatics, we try to find out both the type and also the abundance of microorganisms that are there at a given time, at a given site. Then, of course, proteomics and metabolomics came into being. This is where people started with development of technology to look at the protein composition of microbial origin and, of course, of human origin also at a given site at a given time. And then metabolomics actually revolutionized this field even further. Metabolomics is where we actually study the small molecules that are present in any type of biological sample that is taken from a side. So we will come back to metabolomics as we progress through the lecture. So now originally we were talking about a single pathogen posing a single infection. This is the classical Koch postulates that we all know about, that to establish the etiology of an infectious disease, we have to isolate it from a person who has got it, inject it to some animals or people who don't have the infection, get the same infection, and then re-isolate the bug. This is still valid even as of 2019 when COVID-19 came into being. The classical Koch postulates were followed to a certain extent to establish the etiology being SARS-CoV-2. However, with the understanding that, there are a lot of microorganisms living in us at a given site, and that they actually, there is a community of them at a given body site at a given time. Oops, sorry about it. There is a little problem. Right, I hope the slides are visible again. Sorry about the interruption. So we thought, or rather not us, it's actually the scientists who are originally starting to work on this, the concept that these microorganisms that are occupying a given body site behaved as an organ on its own came into being. So most of the work was done on the gut normal flora, as we called it back then. The gut normal flora became the gut microbiome. So most of the studies were originally based on the gut microbiome. So at healthy state, there is a given abundance and diversity of the microorganism in the gut, and that is known as a eubiotic or eubiosis state. And then when there is an illness, there is loss of diversity as well as a change in abundance of the gut microbiome. There is a dysbiosis. And when there is dysbiosis, this is associated with disease. This is the thinking behind microbiome acting as an organ in our body. So the initial microbiome studies, when they started, were actually just aimed at characterizing the different microorganisms present at a given site by sequencing. So I just did a PubMed search using only the word microbiome. There's over 100,000 studies there. And you can see how exponentially the number of publications have increased over time. Then people moved from just characterizing like normal healthy individuals to characterizing the microbiome in patients with different disease groups. So there we started to identify associations that healthy human microbiome differed from the microbiome of different people who have got different diseases. So this came slightly behind the initial characterization of the healthy microbiome, and then this became into like association studies. So there are plenty of research groups who started working on the gut microbiome, mostly of those with different non-communicable diseases, be it metabolic diseases, skin diseases, even central nervous system related diseases. And they started to identify that the gut microbiome in these illnesses differed from what was considered to be the healthy gut microbiome. So therefore, the original concept in physiology that we all learned about the gut-brain axis got another player into it, the microbiome. The gut microbiome actually plays a role in it. And in addition to the brain-directed activity, a strong link was established with the gut microbiome and the immune system. And of course, a strong link was also established with the gut microbiome and metabolic disorders. Right, so now I'm going to take an example and maybe try and see how the microbiome studies evolved over the time. So I'm going to take a very common example of a non-communicable disease, which is diabetes, and how microbiome studies evolved in this disease. So initially, people started to characterize the gut microbiome in patients with diabetes in different target populations. I know it's very difficult to read the slides here, nearly impossible, but these are our initial studies from the early 2000s, and the sample sizes are like 20, 30 or 50 patients and some age-matched controls, where they started to identify that the microbiome composition and abundance changed, is what's different. Now, after this characterization studies, people move towards association studies, where they actually try to put some statistics, move toward larger patient sizes to actually see whether there are significant associations between these different groups. So this is a very recent study just to show that things are still happening in the same direction. But if you can check, they've characterized the microbiome in 5,572 adults. So now this is a lot of work, very data-rich studies. So these are now being analyzed to identify the specific associations. Now, in addition to just correlating the associations of diabetes with microbiome, then people started to characterize to see whether the microbiome can actually explain some of the complications in diabetes, like the kidney diseases, like retinopathy, and any other complications that we get with diabetes. So again, when sub-analyzing the different groups of people with diabetes, they started to identify that microbiome differed from, like, say, someone who is simply having diabetes when it's compared with someone having diabetes plus a complication. Of course, then, now there are plenty of studies where they have actually identified that there are differences in the microbiome, different compositions, certain pathogenic strains are higher in the microbiome in people with certain complications. So this is now well established, and they have also done some experimental studies where models like fish or mice models have been used, where the microbiome taken from different people with different complications have been injected into these germ-free animals, and they've replicated their to a certain extent. In certain studies, they've been able to bring about the diseased phenotype by giving this microbiome to those animals. Now, of course, we know there's an association, but what actually drives this association? That is the next question that people working on microbiome set about to answer. Now, I initially started, like, briefly introduced to the concept of the metabolome. This is where metabolomics came into be, where the small molecules that are, produced by the human body or by the bacteria, or as an effect of the bacterial presence by the human body, will started to be characterized. And there they identified that certain metabolites of either human or animal origin are implicated in these changes in the microbiome. And these metabolites are in fact the ones that are driving the actual change. The microbiome initiates it, but metabolites go and they are like the, they actually do the work for the microbiome. And in addition to that, they've also now sort of working on this line, some of these metabolites themselves and also the presence of some of the microorganisms, in fact, change the way some protein expression and nucleic acid synthesis processes also in the human gut. So that is how the crosstalk happens between the microbiome and the human through these metabolites by different mechanisms involving changes into the protein synthesis pathway. So coming back to diabetes, the same thing has been applied to diabetes, where they have identified bile acids, short chain fatty acids, branch chain amino acids, and a couple of other compounds are the ones that actually drive the change which is associated with the microbiome changes. And all these metabolites are meant to create a low-grade inflammation in the gut, and also, contribute to insulin resistance. Now, of course, if the microbiome is associated with diabetes, what is going to happen to it with treatment? So the treatment microbiome interaction is a bidirectional one. Certain microbiome compositions actually affect the drug to start with, so that the drug metabolism is interfered with and the control of the disease is not optimal. And in addition to that, people have also identified that with treatment, the gut microbiome reverts back to what is called a healthy microbiome. Now, findings are not 100% conclusive. However, there are plenty of studies that are actually going on now, not only for diabetes, but for other studies also, where they are looking at how the microbiome is changed with treatment. And now people are studying it in further sort of ways where they're looking at, now there's a lot of market for natural products in terms of control of non-communicable diseases. Now they're looking to see if these natural products are used, whether the gut microbiome in diabetes can be made healthy. So there are some studies with certain extracts in relation to gut microbiome in animal models and also in patients. However, it is still a correlation. There are attempts to prove causation. Is it the change in the microbiome that leads to diabetes, or is it the diabetes that leads to the change in the microbiome? Yet to be conclusively answered. However, we know for sure that there is some association there. And of course, this association is not something uniform. There are a lot of individual variations to it. Right? So it is still an evolving field, but it's a field that we need to keep an eye on. So what about other diseases and gut microbiome? There are a lot of evidence to say that gut microbiome is associated with metabolic diseases, obesity, and a lot of other diseases. It's not just the gut microbiome and metabolic diseases, but gut microbiome is supposed to be now associated with all the diseases under the sun. So there is brain, like psychiatric illnesses, organic brain diseases, endocrine diseases, GI diseases, lung diseases, liver things, obviously, bone diseases, muscle diseases, reproductive system associated diseases. People have identified that gut microbiome in anyone with these diseases differ from what is known as the healthy gut microbiome. So this has sort of started to drive off another side of this, like discovery, where people are now trying to see if lifestyle modifications are done, particularly for non-communicable diseases, whether this is able to revert the gut microbiome to a so-called healthy status. And they have actually found evidence for that. Now, the much touted Mediterranean diet is supposed to improve the gut microbiome. There is obviously more room to go, like further things to be explored, but these findings are now coming to the light. And of course, exercise is supposed to modify the gut microbiota also. And it is not only the gut microbiota or gut microbiome, that is associated with disease, the oral microbiome, the lung and the respiratory microbiome, and the skin microbiome. There is evidence to say that all these microbiomes are associated in some way or the other with different disease entities. There are changes in the composition, the abundance of microorganisms. These changes lead to certain metabolic products being released, and they are going and contributing to pathogenesis of these diseases. Now, of course, we have to take these studies with caution, and when you're reading a microbiome study, we have to be always mindful of certain things, because there are plenty of evidence coming out, urging us to exercise caution also. Because some of the earlier, now this involves a lot of bioinformatics, some of the initial analysis bioinformatics systems were found to have errors, and also on the lab side, everything has microorganisms. So unless we take extreme precautions, environmental contaminants can completely change the picture of a microbiome study. So using proper controls and standardizing the procedure is extremely important for any microbiome study. However, having said all of that, microbiome-directed therapy is actually being practiced, particularly fecal microbiota transplantation. This is where the healthy poop of a person is taken out and it's cleaned in some ways, and then it is transplanted in different ways to someone who has got an illness. This was originally trialed and is in regular use for diarrhea associated with Clostridium difficile. But in addition to that, there are active clinical trials going on where they're trying to see if fecal microbiota biome transplantation can have some effect on certain metabolic, psychiatric, neurological, autoimmune diseases. So the clinical trials for these are ongoing. And in addition to fecal microbiome transplantation, there are other studies also, other therapeutic interventions which direct to either try and manipulate the microbiome, say trying to kill off certain unhealthy microbiome bacteria in the microbiome with phage therapy, giving diet. So that arm of therapeutics are also being investigated. And on the other side, they're also trying to see if the metabolomics can be manipulated by giving the actual intermediates of a so-called healthy microbiome. Now, these are brought about reset of request the
Those who are joining online, please actually mute your mic.
Okay, sorry about that. So we already know about prebiotics and probiotics, and our acting dean for today, I'm sure, is very fond of those. But now there are more things coming into play. There are symbiotics, postbiotics, nutribiotics, and pharmacobiotics. They all have different combinations of live microorganisms and things that are needed for these live microorganisms to grow. And of course, postbiotics are the metabolic compounds of the healthy microbiome. So these are the terms that are now coming into therapeutics field in relation to the microbiome. And of course, even though there are still trials going on, the pharmaceutical industry and the other industries have already taken the clue, and they are using microbiome in marketing. So for a lot of skincare products and all, they are already talking about microbiome friendly products. So that is how the association between microbiome and non-communicable diseases are being now, the mystery is being solved. There's yet a lot to be identified, but that side is being investigated. But on the other side, the microbiome studies are also starting to question some key concepts that we held true in medicine. For one thing, whether there are actual sterile sites in the body. That is a question now being asked. People have identified that there are microorganisms in the bloodstream. Of course, it was known earlier also there is transient bacteremia associated with different things. But is there a blood microbiome that is associated with certain diseases? And do we have a synovial fluid microbiome? Any other sterile sites? Is there a microbiome? Is there a brain microbiome? So certain evidence is emerging, and they are starting to question whether there are any actual sterile sites in the body. And this is incidentally where I worked on for 7 months during a visiting sort of a research associateship that I held, where we worked on the blood microbiome and synovial fluid microbiome signatures in patients with rheumatoid arthritis. Also, now we are starting to question whether non-communicable diseases are actually non-communicable, or whether there is a communicable arm in the non-communicable diseases. A very recent study actually found something extremely interesting. They found that gut microbiome is usually transmitted from the mother, vertical transmission, and it's stable, whereas the oral microbiome has signatures of horizontal transmission. That is, household members share similar oral microbiomes. And these authors conclude, I quote, by saying the extent of microorganism transmission between the described underscores its relevance in human microbiome studies, especially the non-infection microbiome associated diseases. So, there is a lot of things to be explored in this area. So in addition to that, we are also trying to see, they are finding evidence to say that the gut-brain axis influences the cognition and drives certain other behaviors like binge drinking also. So, are we capable of controlling ourselves 100%, or are the microorganisms in us controlling us in some way? Now, coming back to tropical diseases, because we are meant to talk about tropical diseases. The microbiome in tropical diseases the microbiome has an impact on tropical diseases. Studies have identified that gut dysbiosis has a role in the progression of dengue to severe dengue. And of course, gut microbiome and different types of malaria involvements. And there's also a lot of talk about the gut microbiota involvement in leptospirosis and things. So these are only tropical infectious diseases. And on the other hand, there's a lot of microbiome studies going on in the world in relation to chronic kidney diseases and the impact of the microbiome on that. Let's come back to a fundamental sort of area here. Now, from the initial primates throughout evolution, we've had a microbiome. So this microbiome is actually evolving with us. We are evolving, the microbiome is inside us, and the microbiome is evolving. The microbiome is actually called the intimate traveling companion of the humans. So a human is now, some people call it human holobiont. That is, we are a collection of a complex life form, not only formed of human cells, but human cells and microbial cells. So there is transmission of this and we are co-evolving. However, this co-evolution is supposed to happen over millennia. With the advent of medical technologies, we have interventions like diabetes and our lifestyles change, and these have a profound impact on our microbiome. Not so much on the human evolution, but they are changing the evolution of the microbiome immensely during a very short period of time. You can see this clearly in migratory communities. So migration from say different areas to a Western country is known to change the microbiome in people, and this has been identified as to have an impact on the metabolic syndromes that are increasingly seen in the migratory communities in Western countries. So there are some interesting groups who are working on this. They've actually studied the microbiome in indigenous communities. And what they have found is that the microbiome of indigenous communities, irrespective of whether they are from the Amazon, in Java, or somewhere else, the taxa there, the microbial composition is quite similar, whereas in industrialized countries, the microbial composition is similar, irrespective of where they are. So this has sort of led to this hypothesis that the disintegration of the evolution of the human genome and the microbiome is actually responsible for most the pandemic of non-communicable diseases that we are seeing right now. So I have linked an interesting YouTube video where they talk about this in the American Society for Microbiology lectures, and if you can spend some time, it is worthwhile listening to that because they explain how the experimental evidence for this hypothesis. So in conclusion, microbiome studies are we can't ignore the microbiome anyway. It's here to stay with all the technology that came. There are definitely associations with microbiome with a lot of infectious diseases like tropical diseases and non-communicable diseases, but causation still remains to be proved. This is also redefining being human in a way. Are we really a human? What sort of impact does our microbiome has on us? Are we actually a working ecosystem? And are we actually being controlled like a puppet by the microbiome that is living in us. So whatever it is, as healthcare professionals, this is an area that is developing very, very fast, and there are therapeutic implications of it and interventions coming into be. Therefore, we have to keep updated about the microbiome related area in our practices also. Thank you, and I would like to thank the CRTM staff, Professor Kularatna, the director, for inviting me once again. Thank you.
Thank you very much, Veranja, for that very interesting and detailed lecture. I mean, you made it very clear to us one of the gray area that we are in. So I hope that you would answer some of the questions the audience has, both online and present here. Any questions you have? Yes, yeah.
Yes, of course.
So thank you, Veranja. So it was a nice presentation. My question is on, again, as you said, probiotics. Are they always helpful? Because we see a lot of probiotics being used for pediatric medications, but we always have this dilemma whether are they really useful, because sometimes can it be harmful as well?
So this is like, say, my answer is based on what I have read, obviously. So according to what I have read, the jury is still out there. So evidence is split on it. For certain things, they have found it's helpful, but for certain other things, they have not found. And even for things like, maybe diarrhea and all, again, when you actually dive into the literature, the evidence is not conclusive. Yeah.
And about there was a recently there was a search on these postbiotics. They thought postbiotics are much better than probiotics because we are not using any live organism. So are they safer than probiotics or still the same?
I haven't actually read into that much in detail.
Because the research went up and went down the same rate it went up.
Yeah. So
because it looks in theory it looks nice. You are using only the products but not the organism. I don't know how stable they are.
Yeah, they probably might not be stable. So there's a group in Stanford who are working on a similar line, but differently. They're trialing with diet modifications where they've tested giving prebiotics, probiotics, and postbiotics and compared it with actual dietary changes where they have given in like a more fiber-rich diet and fermented diet and all. So what they've identified is like, according to their results so far, it's the fermented food which seems to have had a stabler effect. So that's from my reading. Yeah.
Any other questions? Yeah. I would like to ask a question from Chair. When actually, is there something like mapping of microbiome in the globe, and what is the status currently?
Yes, so there is a project, the human, just like the Human Genome Project, there's a Human Microbiome Project. So they are mapping in different areas. Now, so again, this is being questioned now. They're saying they have sort of defined a healthy microbiome, but now they're saying that healthy microbiome is that the actual healthy microbiome? So they're characterizing but questioning the definitions and things. Ongoing work.
Right. Thank you, Veranja. Sure.
Yes. I haven't read.
Just I see now, as you said, in some families, certain diseases are more common, although they are not communicable, right? I think even emotional things are more common in some families. So whether it could be due to these things, maybe the diet and all those, their habits also could be similar, no, in the same. Yeah, and also this sterile syndrome, unlike when we were children, we used to play a lot with dirt and, there was no this concept of sterility. But I do find now the new generation, they have a lot of allergies and a lot of these things could be all this.
True, madam. This is another line that people are studying. So they're saying the changes in the two microbiomes, that is the microbiome within us and also changes in our interaction with the environmental microbiome, whether both of those have now contributed through, they're calling the current status as an inflammasome status, where there is ongoing myoclonic inflammation in everyone, which is contributing to NCDs as well as the immunological diseases like allergies and all. So, as you said, madam, those are being studied, I think.
Any more questions? I don't know whether you have time. Yes. Anyway, we have taken almost 45 minutes. On behalf of CRPM, I should thank Veranja for giving this very interesting talk. And actually, it has opened a big area for research. Actually, I can't think, the limit of it, right? So this young generation can take up a lot of work on it and contribute to the knowledge, right? So what we think today may be different in another few years' time, right? A few months' time. Okay, so ladies and gentlemen, this activity once a week, once a month lectures will be conducted in future, and all these lectures are recorded and will be available in the YouTube for anybody to listen later. So, Chamar, thank you very much for organizing. Also, I thank our acting dean here, right, for your presence. Right. Once again, thank you very much.
Thank you.
Thank you.